Bon dia a tothom, bon dia als que esteu aquí, a tots i totes, i als que esteu també en streaming. Com sabeu, passem a gaudir ara de les presentacions, de les publicacions més votades per tots nosaltres, el 2020 i el 2021. Tenim dues publicacions, farem les dues publicacions seguides, les presentacions, i després així seurem i podrem tenir una miqueta de discussió si tenim preguntes. La primera ens la presentarà l'Stefan Humer, és del grup del doctor Santiago Ramon i Cajal, el grup de patologia molecular translacional de l'àrea d'oncologia del VIR, i ens parlarà d'integrines i de tràfic intracel·lular, i el títol de la xerrada és ITVG, Mediated Uptake of Small Extracellular Vesicles Facilitates Intracellular Communication in Breast Cancer Cells. So, Stefan, you can talk about 15 minutes, pots parlar uns 15 minuts i després 15-20 minuts, no t'allarguis massa, i després seiem i deixem pas al Gerard que ens faci la segona xerrada i després discutim tots plegats una mica, si us sembla.
Well, I better speak in English. I could do it in Spanish, but I think then you would love the same than before, so I prefer to do it in English. Thanks a lot for the organizers for these jornadas. Again, a very nice meeting of the Institute. Also for the PIs that have voted our publication. The publication is done together in the group with Santiago Ramon y Cajal. We co-directed this project. And I want to tell you in very brief what are the main outlines or what were our main findings and why we believe it deserved to arrive to Nature Communications paper. Breast cancer is a very prevalent case of cancer. 1 out of 8 women will finally develop breast cancer. And the cure has hugely increased over the last years, and as soon as the disease is detected early, it's very good chances of treatment and survival of the patients, but once there is distant metastasis, the survival rates drop dramatically, and therefore, like in breast cancer and in all other cancers, metastasis remains the major cause of death, and there is currently it's almost an unmet clinical need. So there is really a lot to do still to understand better how metastasis works and why we still are not able to really treat this disease. Metastasis starts from the invasion of the primary tumor site. The cells invade into the surrounding environment. They intravasate finally into the circulation. The cells float in the circulation. They have to survive all these steps. They extravasate at a distant site, the lung, the brain, wherever the metastasis arrives finally. The cells need to form a so-called premetastatic niche, so they need to engage in a new environment. They start growing and they form finally metastatic colonies. In our lab, we are interested in a protein called ITGB3 integrins. These proteins, just to briefly explain you, they come in the following flavor. They have an alpha and a beta subunit, and the two proteins together are placed on the cell surface and are inserted in the membrane. These integrins are in two forms. Either they are inactive. Once they are getting activated, they mediate what was studied for the last 50 years almost. They engage into the extracellular matrix, so they attach or they provide the interaction of the cell with its surrounding. And once they are activated, they also process a couple of different signaling pathways to drive different processes within the cells. We got interested in the role of the integrin beta-3 in a publication from 2017 from the lab of Santiago, where we identified the integrin beta-3 in a screen for novel metastatic or tumor malignant factors. And the key finding of this publication was that when we take away the integrin by shRNA-mediated knockdown, the total number of lung metastases in a tail vein injection model, so what we do here is we inject the cells in the tail of the mice. The cells arrive directly to the lung, and we can measure the metastases in the lung. It's a fast way of determining cancer metastasis, and this is largely reduced if we take away the integrin. So for us, the question was now, since we injected the cells into the tail, they enter directly in the lung. In which of these steps is the integrin actually important? And as I told you before, many people were thinking in the role of integrins as attachment factors. So we also thought, okay, let's try this. Let's see if when we inject the cells again in the tail vein and we harvest the cells relatively fast after after an overnight inoculation, do these cells really stay in the lung? So do they attach or do they not attach? Surprisingly, there was no difference between wild-type control cells and the knockdown cells. So the cells obviously are still able to arrive to the lung, to attach to the tissue, but then the consequence was that they still don't form metastasis. So what is going on? And we focused then on the second step in this cascade, which is the colony formation. In this process, there is an important role of the intercellular communication between the tumoral cells and the cells in this environment. One can imagine this as the tumor cells have to be able to communicate with all these cells that are there, because these cells either hold metastatic formation or they promote. So they have to find a way to communicate positively with these cells. And the factors we are mostly interested in this regard, or what we found, are so-called extracellular vesicles. I don't want to go too much into extracellular vesicles. Just to give you a brief overview, we have different types of extracellular vesicles, the ones that are shed directly from the membrane, or so-called exosomes that are generated in the cells and secreted. There's a very heterogeneous population of vesicles. They are different in size, in their content, and we are only just starting to understand what these extracellular vesicles do. What is absolutely clear is that they promote metastasis. And so to set up an assay on testing this colony formation potential, we used the following conditions. So we used we used a so-called colony assay. You see the very low number of cells. And we supplemented it with so-called conditioned media. Conditioned media is nothing else than old media, or 24, 48 hours old media, that is rich in factors secreted by the other cells. Actually, this assay is adapted from whoever has ever generated a clonal cell line uses exactly this trick, to use old medium to grow the cells. And what we saw is that if we supplement wild-type cells with conditioned media, they grow much faster than without this conditioned media. In contrast, the integrin knockdown cells did not grow at all. So they did not, or they grew somehow, but much reduced. So they are not able to react to what is present in this media. And even more, if we take out now from this conditioned media only the extracellular vesicles, the cells are not able to respond. So it's the extracellular vesicle part that promotes this growth. Just to clarify, it is really important to have the role of the integrin within the cells, because if we use this conditioned medium from integrin knockdown cells, we see the same effect. So it doesn't matter if the integrin is present in the vesicles or not. It is important if it is in the cell or not. And finally, to make this more relevant for the metastasis, IMR90 is a fibroblast cell line that is derived from the lung. So even normal lung cells are able to stimulate this growth. So in a way, the cells are able to communicate in the lung environment with the tumor cells, with the stromal cells in the lung environment. Well, this then we used biochemistry to understand exactly the mechanism of this process. What we basically saw is that wild-type cells normally take up extracellular vesicles in a time-dependent manner. The IgG3 knockdown cells are not able to do that, or much slower. And again, this doesn't depend on if it's the vesicles are derived from the wild-type cells or the integrin cells. So it's the integrin in the cells. And how does this work? I don't want to go too much into detail. For the people that have heard about the focal adhesion complex, this is a complex that fixes the cells or helps the cells to attach to the surface. There are several important factors, and what we have found is that actually the integrin that is placed here, which is recycled over time, during all these factors that are important in this recycling process, like dynamine, like FAC, a kinase that is important for this process, they all are able to mimic this effect. So we have here the reduction in the vesicle uptake with integrin. In green, we have the uptake defect with a dynamine inhibitor. An unrelated inhibitor does not give us any effect. The same, again, with the knockdown of FAC by siRNA. And most interestingly is that it was already known before that heparin is able to block extracellular vesicle uptake. So we were wondering, are heparin binding proteins, so-called HSPGs, are they important in this process, and do they cooperate with the integrin? And the answer is yes, because if we knock down the integrin and treat the cells, if we treat the cells with heparin, we see a knockdown. But if we treat integrin knockdown cells with heparin, we don't see an increase in the reduction, implying that this is a complex of heparin binding proteins and integrins that is responsible for the uptake of these extracellular vesicles. Here on the right side, this is another way of reducing the heparin binding activity, is to use heparinase and to cleave off these side chains from the heparin binding proteins. And finally, we could reveal that this is actually all linked to the activity of this kinase called FAC. If we incubate the cells with extracellular vesicles, the activity of this kinase is increased, and this is dependent on the integrin, on the dynamine, all the factors that I explained you before. And we came up with this molecular model on how basically the uptake of the extracellular vesicles work. The vesicles first bind to these so-called heparin binding proteins, the HSPGs. If we treat with heparinase or heparin, we block this binding. Then we have an association of these heparin binding proteins together with the integrin. Again, if we block the integrin, we stop this association here. This together activates the focal adhesion kinase, which results in the internalization of this co-complex into the cells. And exactly in these ways, the vesicles are internalized. So they basically hook up on an existing mechanism to enter into the cells. And since this was important to have the molecular details defined, now for us working here in the hospital, of course, the big question is how can we address this clinically? And basically focusing on how we can prevent the metastatic colonization. And well, I don't have the time now. Maybe next year, if you invite me again, I'll present you what came out from the studies. we have a poster on this, but we have studied several different inhibitors, heparin binding inhibitors, integrin inhibitors, function blocking antibodies. We are producing nanobodies as a tool to prevent this. And so far, we have posted this here with a new heparin binding mimetic protein that is very efficient blocking the extracellular vesicle uptake, and we are testing it now in mice models if this can indeed prevent metastasis. And finally, to show how this project can be also translated into other clinical fields, it's surprising, but if you look at the diameter of extracellular vesicles and a virus called SARS-CoV-2, they're actually pretty similar in size, and also the surface proteins are relatively similar. And I have to admit that we have learned a lot of the basic concepts of our studies from comparing the role of the integrin in viral uptake. So it's not that we invented it. It was already known that the integrin has a role. And so teaming up with Maria Jose Puzon from the infectious disease group, we basically asked the question, if we deplete the integrin, do we get a defect in the uptake of SARS-CoV-2? And this is indeed the case. So we take away the integrin, and if we use a construct that has the viral spike protein, we see a clear reduction in the uptake of the SARS-CoV-2 pseudovirus. If we use a control that does not have the spike protein from SARS-CoV-2, this doesn't happen. Why is this? There is maybe one easy explanation, and that is that SARS-CoV-2 has developed a so-called RGD motif, which is the classical integrin binding motif that SARS-CoV-1 does not have. So the integrin binding motif might be one of the parts why this virus is so much more infectious than its previous version. We keep on collaborating with Maria Jose Puzon in a way that we develop our inhibitors anyway for metastasis, and whatever we find that is effective, we are testing in parallel also for the uptake of SARS-CoV-2. And I think this is a nice way of bringing basic and clinical science for two questions together. Well, that is basically all. My special thanks to Santiago that gave me also the opportunity to talk today, and for all his support and for studying a bit of more molecular biology, but I think it's important to understand the processes before translating them into the clinics. And thank you very much for your attention.
Thanks, Stefan. Very nice, very clear. Very nice example of what you said. So bringing together different expertises and disciplines to better understand progression, in this case of breast tumors and also the COVID disease. Passem a la següent, si vols seure, Stefan, i després fem preguntes i comentem. Passem a la segona presentació, que la farà el doctor Gerard Cantero, del grup de l'Anna Massaguer. Ens parlarà de la malaltia de Dent. El títol de la seva xerrada és el grup de fisiologia renal, fisiopatologia renal, i la publicació, veu poder publicar aquesta feina a Human Molecular Genetics. I el títol de la presentació és Novel Dent Disease 1 Cellular Models Reveal Biological Processes Underlying CIC5 Loss of Function. O sigui que, Gerard, quan vulguis.
Perfecte. Hi. Good morning, everybody. I will also speak in English for all of you. First of all, thank you for this nice introduction. As she said, I'm a junior PI of the Renal Physiopathology Group, led by Dr. Massaguer. And, well, yeah, first of all, I also want to thank the organizers for inviting me to present our results here and for our manuscript. So today, I will talk about our results that we published in the Human Molecular Genetics about novel new cellular models for Dent's disease, that is a very rare renal disease, And first of all, I want to start with the very, very, very beginning. So the kidneys. Most of you for sure know that they exist. Maybe you even know the shape that it has, that it's this shape. Maybe you even know the localization, but very often we don't realize how important they are for life. Indeed, they are essential for optimal body homeostasis, and amongst others, the functions that the kidneys have is blood filtration, that you already know, but also regulation of electrolytes, osmolarity, maintenance of the fluid, the volume of the body fluid, acid-base balance, production of several hormones, and this is very important for our body, is the excretion of wastes and toxins. The main functional unit that realizes that does these functions is called the nephron. Here you have the drawing of the nephron that is basically the functional unit of the kidney, and is made of a filter, that is the glomerulus, attached to a tubule. So this glomerulus filtrates the blood, and as you can imagine, a lot of substances will pass through this filter, like many of them you need to recover because you don't want to waste water or ions through the urine. So one of these very important parts of the nephron is called the tubule. The tubule can be divided between the distal and the proximal tubule. And today, because its importance in the disease, I will focus on this, on the proximal tubule. This part of the nephron is involved in the reuptake of water, of small molecules like ions, for example, calcium or magnesium, for example, and also low molecular weight proteins. This will be very important later on for the disease that I will talk to you. This disease called dense disease. The cells that perform this function is the epithelia of this tubule, and these epithelial cells, they have a very specialized apical endocytic machinery that is the one that will reuptake these molecules from the medium and endocyte them to the intracellular part of the cell, and then they will be delivered to lysosomes, or depending on the ligands and the receptors, they will go to other parts of the cell. So a very important step here is that we need some receptors or some molecules that will bind to the ligands, and these ligands will then be endocyted and delivered to the lysosomes. But a very important step here is that the receptors need to be recycled back to the membrane so they can continue this cycle. A very important element here is CLC5. I really like this molecule because of my background in channelopathies, and this is, it's not a channel, but it's an antiporter of chloride for protons. What it does basically is to exchange chlorides for protons. It's localized at, as you can see here it's in yellow, but here it's the structure of this antiporter. So it's localized at the plasma membrane, but mainly in these apical endosomes where it can control and regulate the pH. By regulating the pH, it may have a function in the maturation of the endosomes, but also in the recycling of receptors to the plasma membrane. So this is in normal situation, but what happens when this channel, this antiporter, is not working correctly? What we know is that mutation or loss of function of this channel leads to a decrease in the endocytosis and in the number of receptors. This, here, so many years ago, Dr. Dent identified a disease, described for the first time a novel disease, that also had one of the hallmarks of this disease was low molecular weight proteinuria. That is one of the hallmarks of tubule dysfunction. It was interesting to find years later that this disease was caused by mutations in this gene. So, and this disease is also characterized by hypercalciuria, nephrolithiasis, nephrocalcinosis, and what's very interesting for me and for what our group want to study in the future is that this disease progresses to renal fibrosis, requiring renal replacement therapy. What's really important also here that this is a very, very rare disease. It's only 300-400 families worldwide, and more than 200 mutations has been described. Every mutation, what they have in common is that they cause this loss of function of this gene. And what we have right now is that there is no treatment for this disease. So the main question that we want to solve is what cellular mechanisms link this protein with proximal tubule dysfunction, because what we know is that if we have these mutations, what these mutations will produce is a decrease in the endocytosis, in the recycling of these endocytic receptors, but this somehow leads to a decrease or cell dedifferentiation, and altogether this will trigger this dysfunction of the tubule that will end with all these characteristics that we see. So even that we know, or for many years it was known, the genetic cause and the phenotype, the link between the two wasn't clear. So that's what, in order to solve this question, in our lab, what we decided to do is, first of all, to generate a novel cellular models for the disease that could explain this link. So this is the paper that we published. This is, that is led by Dr. Massaguer, and it was a very simple strategy. So first of all, we used RPTEC, that are renal proximal epithelial tubule cells, and in these cells that they express the CLC5, first of all, we silence the gene, and then we rescue the phenotype with including different constructs with the wild-type form of the CLC5 or the mutants of this CLC5. Here I show you the structure, and what we did, we used the wild-type form of the CLC5, but also mutants, that all these mutations are in the same loop, but they have different effects. One of these mutations is very interesting for us because it's from a patient here at Vall. Another one, it's a mutation that it doesn't cause any difference in the location of the protein, but it has an effect on the function. And finally, another mutation that it affects both the location and the function. Obviously, in order to prevent this entry of the wild type or the mutated forms to be affected by the SH, we included some mutations so these constructs are not affected by the SH. So first of all, we needed to characterize these cells, and what we found is that really the SH is really knocked down the endogenous CLC5, and then when we introduce the mutants or the wild type, we have an overexpression of the CLC5. What's here, we were really smart, and what we did was to include a tag, so it would be very easy to detect these genes. Here you can see that after when we introduce the wild type, we have overexpression, and we already can see some kind of effect when we overexpress the wild type. So there is some defect on the expression. This can be because of problems in the synthesis or because of enhanced degradation. Here I show you the normal distribution of the wild type CLC5 that is at the plasma membrane, but also in some vesicles that maybe they are endosomes. I will show you later that, yes, they are endosomes. And when we overexpress these mutants, for example, this is one of the mutants, it is retained intracellularly and it never reaches the membrane. So we really characterized this, and what we found is that here you have the wild-type form and then the different mutants, and what you can see already in the pictures here is the CLC5 staining. I only show you one of the stainings we did because we did many, many experiments. In this case, we co-stain it with RAP5, which is a marker of early endosomes, and you can already see that the wild type really reaches the plasma membrane, and it also is in these structures that now we know that are endosomes, but the other mutants, they are retained intracellularly, or for example, this one that I told you that it can reach the endosomes, more or less. There is some of the RAP5. Oh, sorry. So here you have the quantification. You can see that in the case, so I start by the end, that the wild type goes to the plasma membrane while the mutants never reach the plasma membrane. When we compare the colocalization with the endosomes, we see that the wild type really colocalizes. The other mutants don't colocalize, but the one in the middle, that is this mutant, has more or less some colocalization with the endosomes. And then finally, we have the colocalization with the ER, that is, will be a marker of the intracellular retention. We have that one of the mutants that is known to be retained in the ER is the one that have the highest colocalization. So as a conclusion, mutations in the CLC5 cause this mislocalization of CLC5 to some intercellular compartments. And this could explain the lack of function. So one of the characteristics of the disease is that there is a lack of endocytosis. So first of all, if we want to really know if our cellular models are really replicating the disease, we needed to test if endocytosis was affected in these cells. And that's what we saw. So this is a very satisfying for us. Here we were also, what we did was to test endocytosis by incubating the cells with the albumin, and then we measure if this albumin can be integrated by cells or not. You can see here is the 3D projections. In green is the albumin, and in blue are the nucleus. So here you have a orthogonal view with in dashed line is the membrane. You can see that in control cells or in wild-type cells, intracellularly we can detect this albumin. But this is totally avoided in the knockdown cells. And here you have the quantification, not only for the knockdown, but also for the mutants. So what we found is that the loss of function of CLC5 by knockdown or by mutations causes defects on the endocytosis, and this is recovered by the wild-type form. So at this point, what we found is that our cellular models really work and can replicate the phenotype of the disease. Next, what we did is to test what genes were altered in our cellular models. So you can already see that there is a huge difference between the control situation and when we knock down the cells. These are microarrays of the RNA. And then when we overexpress the wild-type form, we partially recover the expression of the control. And here you have another comparison that would be the wild-type compared to the different mutants. What's interesting for me is that when we do all these comparisons, we can see that some of the genes went up, others down. But what's really, really interesting is that most of these genes were from pathways related to extracellular matrix organization, wound healing, cellular model, cellular movement, cell-cell adhesion, ion homeostasis, cell substrate adhesion, and all these are hallmarks of a cellular dedifferentiation. And this is what we also see in the disease. So our cellular models really have the same phenotype, endocytosis defects, and when we check what's happening at the gene expression level, we also see this, that the cells are dedifferentiating. So this is at the RNA level, but obviously you know that from the RNA to the protein, we have a gap there. And what we wanted to do here is to really check if there was, for real, the differentiation of the cells when we had the loss of function of CLC5. And that's what you have here. What we tested, first of all, is the epithelial markers that are markers of differentiation. For example, E-cadherin. You can see that this is really striking, that by the knockdown of CLC5, we have almost no expression of E-cadherin. And this is the same with the knockdown, with the mutants, and also with other epithelial markers. So this is pointing directly that there is a problem with this differentiation. And this then we checked because it was one of the pathways that was also altered in our cellular models when we checked the expression. It's the adhesion. And we can see that really cells that have the mutants or have the knockdown of CLC5, they have more adhesion to the substrate. This is also one of the characteristics of the differentiation state. So, because I think it was a bit long, I will go to the summary. What we found here, and that's why our paper is so interesting, is that first of all, we have generated these new dense disease cellular models that can be used to test other strategies or drug to treat the disease somehow. Also, what we found is that the CLC5 loss of function, maybe because of the knockdown or by mutations, causes this reduction in the endocytosis. That the mutations in the CLC5 cause mislocalization of CLC5 from endosomes to other compartments. This is something that we also want to study, why there is this defect, because at the end, these mutations are really close one to the other, and just one amino acid from the other can change this localization from the ER to the lysosome to the Golgi, for example. Then, what we also found is that this loss of function of CLC5 lead to this upregulation or downregulation of different pathways that are related to cellular dedifferentiation. And finally, that this CLC5 lack of function reduces the epithelial markers. But although now we have all this information, there is one question that still remains, that is, how is this CLC5 loss of function cause this differentiation? So this is what we want to study right now, and we have a lot of ideas, a lot of experiments that we are checking. We know now that some of these epithelial markers are affected, but also other secreted proteins like collagens or mucins. So if everything goes well, maybe next year we can explain this to you. And just to finish, I would like to thank, obviously, all the people from the renal physiopathology group, especially what I call the DD team, that is the dense disease team. Obviously, Dr. Masaki for accepting me in the lab and also for allowing me to present this nice story today. Gemma Ariceta, that is also a PI from the group. Monica, that is the main postdoc in the project, and she did most of the experiments. Sonia, that is a master student that recently joined the lab. And obviously, I also want to thank Eduard and Carla, that were important members of the lab and of the group. And yes, sorry, also the funding agencies, especially ASDEN, that is Association for Dense Disease, and without them, this project wouldn't be possible. So thanks all of you also for your attention. And if you have any questions, I will be happy to answer.
Thanks, Gerard. Thank you very much. It was very clear as well. So we try to keep on time. Intentem mantenir-nos a l'hora, però jo crec que tenim 2 minuts per preguntes. Si algú té alguna pregunta. Si no, el doctor Comella sí que en té una. Jo també en tinc una per cada un.
Nice talks, both of you. I have a question for Stefan. You just presented the results on lung cells. Did you check if the mechanism also work for other kind of cells or other kind? I mean, it's related to the vascular tissue or to the final destination tissue. You know what I mean?
Yes. Well, we have not checked. We have focused on lung because it's our model where we see, but obviously, it would be very interesting to, for example, check brain metastasis. But we have not tested. We want to, for example, use now astrocytes and see the interplay between breast cancer tumor cells. We have from Joan Massaguer derived cell line that gives specific metastasis to the brain, and we want to see if they're the same interplay between, in this case, astrocytes, for example.
But what's your idea behind? What do you give?
I believe that is basically this, that the mechanism might be the same. That because at the end of the day, as long as the tumor cells find some exosome secreting cells that can stimulate them, that can maintain them, they will stay there. They only need the signal. They only need the response. And as soon as, at the end, it's an interplay. The tumor cells will probably transform the fibroblasts. The fibroblasts will respond back with vesicle secretion, and then the cascade maintains, and they stay.
So if that is the case, going more deeply on what you are explaining, what will explain the differences between the different places where the metastases are preferentially done?
Well, it's the big question about metastatic homing. There are these cell lines from Joan Massaguer that are supposedly to drive to different places.
I know that you don't have the answer.
I don't have the answer, but the integrin. One explanation at the beginning was that the integrin expression on the but at the beginning it was explained with the integrin expression on the exosomes. I think what one should take into consideration is the integrin expression on the cell surface to test if, because I believe that there are more integrins doing this job, but not all of them. So it will be interesting to define the pattern and to see if this is correlating with the homing into brain, bone, lung. A very interesting question, but yes.
A lot of work ahead. Thank you very much. Congratulations.
More questions? So I have one for Gerard. I wanted to ask you, how do you envision you can translate that into one step further? So where do we go now? Do you have mice models or animal models where you can now restore the function as you were doing with the gain of function experiments and try to see if you can...
Yeah. So what we have now, we have access to samples from patients, and there what we are also checking is the exosomes there, and the microRNAs that are
related.
Yeah, and the microRNAs that can be found there. And with this, we expect to find some of the pathways that can be altered by these microRNAs. And then after that, we want to check what targets we can assess if they are altered also in our cellular models. And the idea is to use our cellular models to test these drugs that can affect these potential targets. So our cellular models at the end can be used for that. You go from the patients, you can obtain some of the possible targets. With these targets, we can identify what drugs could be effective against them. And then instead of going for the animal model or the patients directly, we can first go in our cellular models, and we can do really a screen to test these drugs. So this is what we would.
And is it too naive, or can one imagine or envision some kind of genic therapy with CIC5?
So this is also something
some advanced beyond that?
That would be really nice, because at the end you have seen that with the rescue, only with the wild type, we really rescue
that's why I'm asking.
almost all the functions. So this is also something that we are doing in the lab. Well, other part of the lab is doing with other diseases. So this would be really nice to do. I think that the CLC5, since it's only one gene that causes the disease, it would be a good
target.
Yeah, good targeting for the gene therapy. Although the problem, as usual, for rare diseases is that only very few families are affected, and then always for the pharma companies it's more difficult.
Of course, it's difficult to justify yet.
Yeah.
Okay, thank you very much. I think we'll leave it here. I think they were two very nice examples of how more basic research in the lab can be translated later into good clinical practices. So I thank very much Stefan and Gerard. I think we can finish with an applause to them, and we can move to the next section.
Thank you.